Hey guys, so we're back. This is chapter 2, the chemical level of organization. And so what we're going to do is we're going to identify the basic atomic structure, the major elements, and the difference between ions and atoms. We're going to identify how atoms can bond and the transfer of energy using ATP. We're going to distinguish between organic and inorganic molecules and the biological significance of each group. And we're going to describe respiratory acidosis and then the relationship to the molecules within the body. And don't get nervous, all of A&P is not chemistry and it's not biology. I promise it's going to become its own thing in just a little bit, but we have to get through all of these basics first. So what's an atom? An atom is the smallest unit of matter that's going to retain any of its properties. Elements are substances made of atoms with identical numbers of subatomic parts. What's a subatomic part, you ask? A subatomic part is a neutron, a proton, or an electron. So when we look at, maybe you've seen the periodic table of elements before, and when we look at that, each one has an atomic number, and the atomic number is going to actually help us to determine which type of element is it, and therefore what kind of atom is it going to be. CHON, carbon, hydrogen, oxygen, and nitrogen are the 4 most abundant elements within the human body. And then we already learned what a molecule is from chapter 1, but just to review, a molecule is 2 or more atoms that are joined together by chemical bonds. So there you can see the CHON atoms, oxygen, carbon, hydrogen, and nitrogen. 65% of your body is going to be oxygen, 18.5% is carbon, 9.5% is hydrogen, 3.2% is nitrogen. Okay? We also have calcium, phosphorus, potassium, sulfur, sodium, chlorine, and magnesium, as well as trace elements like boron, chromium, cobalt, copper, fluorine, iodine, iron, manganese, molybdenum, selenium, silicon, tin, vanadium, and zinc. So elements, again, those are made up of atoms, and atoms are composed of their subatomic particles like protons, neutrons, and electrons. Protons and neutrons are going to be found directly within this nucleus. They have mass, and then they dictate the different properties of the atom. Most often on an atom that is neutral, we're going to also have the same number of electrons. Electrons surround the nucleus in clouds or levels. Sometimes they're called shells. Sometimes they're just called valence electrons, and they have negligible mass. They're responsible for the atoms bonding together. These were discovered by Rutherford in 1910. Fun history for you. So these subatomic particles that we're talking about, protons, neutrons, and electrons. Protons have a positive charge. You'll most often see them with a plus symbol here. Neutrons are neutral, so you can use like a 0 or just nothing. And then electrons have a negative charge, so you see a minus sign. So here's that periodic table that I mentioned, and so each one has a symbol on them. So that's usually 2 letters to indicate which, 1 or 2 letters, sometimes 3, to indicate which type of atom or element it is. So here you can see the letter N, that's going to be nitrogen. It has the atomic number 7. That atomic number tells us how many protons nitrogen has. It's going to have 7 protons. If it had different number of protons, it would no longer be nitrogen. Okay. It also is going to tell us that there are 7 neutrons also found within the nucleus, or the center portion. You might hear that word nucleus a couple of times, and we talk about it in different ways. Like we talk about the nucleus of a cell as well as the nucleus of the atom. And nucleus just is that center portion that's sort of containing kind of that most important part. Okay, but we also can have different numbers of electrons. So in a neutral nitrogen atom, we would also have 7 electrons, but we might have 6 electrons or 8 electrons found in nitrogen, and that's okay. So this is those electron shells that I was talking about. So here's the nucleus. That's going to be that center area that's containing the neutrons as well as the protons. Okay, so a positive charge found in here. Okay. And then we have these electrons on the outside. So these electrons are going to orbit or go around them sort of like in a planetary model. There's some idea that it's just sort of a cloud of electrons hanging out there, but they have distinct levels of energy called electron shells. The outer electrons on the outside here, we would consider them valence electrons. And those valence electrons are the way that we can actually bond two atoms together. And we know how many valence electrons are going to be on the outside based on where they fit in the periodic table. Okay, so here, number 1, that means they have 1 valence electron. 2 here, they're going to have 2. 3, 4, 5, 6, 7, and 0 on the outside. Okay, and so these transition metals in the center, they're not really going to be binding in this way, and so we don't have them with those valence electrons. Okay, so the different types of bonds can actually form depending on sort of what's happening with those electrons. So in ionic bonds, okay, we have these electrically charged ions. Okay, so a cation is going to bind to an anion. What does that mean exactly? Well, a cation is a positively charged ion. Okay, what does that mean? That means that we took one of these, let's say sodium. Sodium is right here, Na. Okay? And sodium has just 1 valence electron. And 1 valence electron, not super happy. Okay? Not super happy. So what happens is sodium's like, hey, I don't want this electron hanging out there all by itself. Let me give it to somebody else. Okay. And so if I had an overall negative, or I was just sort of normal and I gave away some of my negativity, what would that mean? That would mean now I'm positive. So sodium gives away some of its negativity, that 1 valence electron, and it's like, hey, you know what? I'm pretty happy now. I'm positive. So sodium becomes a positively charged atom, which is also an ion. And a positively charged ion we call a cation. Okay, right there. Cation has that plus right in there to help us remember that it is positively charged. Okay, now it's a cation bonding to an anion. What is an anion? Well, an anion is negatively charged. So let's look over here at chlorine. Chlorine has 7 valence electrons. Well, you know what? It's really happy when it has 8. Okay? It's like 8 is great. Okay? And since 8 is great and we really want 8, then sodium's like, hey, here you go, chlorine, you can have one of mine. And since you can have one of mine, then chlorine is going to become negatively charged because it got an extra one. Okay, and so cations are going to bond to anions. Okay, so we can donate those electrons out. Okay, so that's how we form that ionic bond, sodium chloride. Covalent bonds actually have a sharing happening, okay? So if you just pretend for a second that I've got a little bit of electron and I don't want to give it to you, but I will share it with you. So we both kind of are holding on to the same electron. And that's how covalent bonds mean. That's co means to share, valent, share the valence electrons. Hydrogen bonds are a little bit different in that they are actually a weak bond, and they are weak because they're relatively weak, so to speak, because it's actually 2 partial opposite electrical charges. So partial charges, meaning that it's just sort of the shape, the way that we've sort of oriented those atoms from one another. And so that actually gives them what's called a pole. Okay? So we have like a North Pole and a South Pole on the Earth kind of idea. And so we get sort of that partial opposite charges. Okay? Water actually bonds with hydrogen bonds. Okay? So let's look at this cation or this ionic bond here. So we use sodium and chloride. Okay, and so the sodium, it's going to become more positively charged because it's going to give away this electron. And now chloride's going to have a net negative charge. So sodium becomes a cation. Chloride becomes an anion, okay, because cations have positive charges and anions have negative charges. And that forms sodium chloride. Sodium chloride, who knows what that is? It is salt. Covalent bonds can happen. We can actually make single or double covalent bonds. okay, depending on what happens. And so this is where we get some sharing of those electrical. So hydrogen gas likes to do this, as well as oxygen gas also likes to bond to what's called a double covalent bond, where there's sharing, okay. Hydrogen bonds, remember, have that weak attraction, and so this is how we form water. So here is oxygen right here, and it's bonded to hydrogen. And see how it sort of orients itself so that we put our hydrogens on one side and our oxygen on the other side. And so that gives us a slight positive hydrogen ion in one molecule and a slight negative here in the same or another molecule. This is a different molecule here. Okay? And so that gives us a relatively weak bond. This helps us to create what's called the 3D structure of proteins. Okay? It also gives us the enzyme active sites that helps us to form a DNA double helix. Hemoglobin forms in that way. Where do we find hemoglobin? We find that within our blood. And hydrogen bonds between water molecules give it a very special property. If you were to put water in a glass and sort of look at the edges of the glass, you can actually see the water kind of creeping up the edges of the glass. We'll find that water actually is attracted to itself. That's how water droplets on the window will actually like form larger droplets. They'll kind of move together. So water, again, it has this polar covalent bond between the oxygen and the two hydrogen atoms that creates that V-shaped molecule here where the oxygen is up higher and then we have the hydrogens off to the one end. It gives us a polar covalent bond. The hydrogen bonds are going to form between the negative and the positive ends of those different water molecules. That gives us this cohesiveness where the water is going to beat up and come closer to each other. It gives us a thermal stability, so it takes a really large temperature change to boil or to freeze. 100 degrees Celsius actually difference, and 0 degrees Celsius is where water is going to freeze. 100 degrees Celsius is where water is going to boil. The chemical reactivity, it acts as a solvent, so other compounds can actually interact within water. And that's really why water is so important to us, right? Because it's providing an environment for chemical reactions to take place. And let's talk about a few of those different chemical reactions. There's a synthesis reaction. Synthesis reactions would be when we're adding together. So this would be an anabolic reaction, right? So we're doing anabolism in synthesis. Okay? So we take two smaller parts and we're building up a larger one. Energy is required, and remember, it's stored within the bond. Decomposition is when we're going to break apart. So if we take that word bookworm, we take it apart, we're going to get smaller products. So we're going to do catabolism here in that decomposition, and we're actually going to release that energy out. We can also do what's called an exchange reaction, where bonds are formed and broken on both sides. Right? And remember, metabolism is the sum of the synthesis or the anabolism and the decomposition or catabolism reactions within the cells. Synthesis reactions are going to combine reactants into products. This requires energy. Remember, we put energy in to store it for later. And we put energy in and store it for later. We call those reactions endergonic. Endergonic. Okay, and we can do what's called a dehydration synthesis reaction. We do those a lot. Dehydration, what does that mean? It means we're going to take the water out, and synthesis means we're going to put two things together. Okay, and so when we make sugars, proteins, and fats, we do dehydration synthesis, and so we pull out water and we can then link those two monomers together. Decomposition reactions are going to break down the compounds into those components. So bonds get broken and it releases energy. And when we release energy, we call that exergonic. Okay? Hydrolysis, that lysis right there, that means to break apart. Hydro means the water. Okay? And so we use water to break down those organic compounds. Same idea. So we add some water in, we break them apart. Okay? So those are exergonic. We also have what are called enzymatic reactions. Our body likes to use enzymes to lower what's called the activation energy to do a reaction. So we're actually able to do a reaction more frequently or even faster because we've lowered the energy required in order to cause the reaction to happen. Okay? And this is like a puzzle. So we have these two enzymes. We have these active sites here. Okay, and we take two substrates. Substrates are just what are we using to make a product. Okay? And we kind of stick them together, and we use this enzyme as sort of the glue to kind of help stick them together. But the enzyme remains completely unchanged, okay? And it fits only specific substrates, okay? And when we do that, that product can detach, the process can repeat, new substrates can come in here and bind. Okay? And so you can see here, this is when we have lowered that energy of activation here for this reaction to happen. Okay? Here, we haven't been able to lower it, and so it requires more energy, and more energy usually means more time in order to get the same amount of products. So we're going to talk a little bit about organic chemistry now. This is a quick organic chemistry. So we're going to talk about peptides. Peptides are proteins, nucleic acids, which is what we found in DNA, carbohydrates, which are fancy sugars, and lipids, which are fancy fats. Okay. And so these organic compounds are always going to contain both carbon and hydrogen. We have both polar and non-polar organic compounds. Polar means that they're going to be water soluble. So carbohydrates, sugar, peptides, which are proteins, and nucleic acids, which are found within our DNA, those are going to be water soluble or polar, meaning that they have a definitive charge on them. And then non-polar is going to be the water insoluble. These are going to be lipids. And you can see here that there's not a definitive charge. It's kind of the charge is evenly distributed instead of having this sort of North Pole idea, which makes it polar. So lipids, if you just think for yourself, these are like fats. So think about some maybe cooking oil or butter. Okay? So lipids are surfactants. That means they're soap-like or slippery, and they have what's called a hydrophobic end. Hydro, again, means water. Phobic means afraid of. So there's an end to them that doesn't like water. They also have a hydrophilic end, and hydrophilic means they do like water. Okay? And so hydrophobic doesn't like water, hydrophilic likes water. And lipids have both a hydrophobic and a hydrophilic end. We have saturated and unsaturated fats, and saturated just means that they are saturated with hydrogens. Unsaturated means that they are not. Okay? And so unsaturated have sort of this bend to them. They have a double bond found. There's 4 types of lipids. We have glycerides, sterols, phospholipids, and prostaglandins. So glycerides, this is the first one we're going to talk about. You maybe have heard of triglycerides. Triglycerides, tri means 3. That's what this picture is. It's 3 glycerol backbones right there. These triglycerides are the primary storage of fat in humans. We actually will check our triglyceride levels as we get a little bit older. Fatty acid chains are these linear chains of carbons. You can see them here. They're attached to that glycerol base molecule here, and they range from about 12 to 24 in humans. Monoglycerides, mono means 1, is a single area right here. That's all we would see as just one of these, is for digestion and lung function. Diglycerides are the most abundant found within our body. They help us to build our body structures like phospholipids. And then triglycerides is the primary storage mechanism of fat in humans. Okay? These are sterols. If you look at that sterol right there, you're going to see cholesterol, right? So cholesterol is the most common sterol found within humans. It's a component of the cell membrane for fluidity. It's a precursor of vitamin D and sex hormones and adrenal hormones. It's a component of bile that we use for digestion and can cause gallstones. It's found only in animal-based foods, and it's synthesized within the liver. Steroids are also part of sterols. So if you think of steroids, that would be like testosterone and estrogen, and cholesterol is the most common sterol. Phospholipids are when we're going to have 2 fatty acid chains and a phosphorus containing group. Okay. These 2 fatty acid chains and a phosphorus containing group are attached to the glycerol backbone. So we have a polar head and a non-polar tail. We also have prostaglandins, and prostaglandins are biologically active phospholipid molecules that regulate many of our physiological functions. We have proper balance of prostaglandins are going to be critical for our normal homeostasis, and we name them PG and then whatever. So PGE, PGFs, okay? And so that stands for prostaglandin, PGIs, right? And they have many different jobs. They help to relax vascular smooth muscle. They help to increase our renal blood flow. They help to contract uterine smooth muscles. They help to relax them. They help to contract our bronchial tubes and also contract. Okay? So lots of different things. These are carbohydrates. Okay, so carbohydrates is another part of these carbon-containing molecules. It's carbon, hydrogen, and oxygen, or CHO molecules. And glycogen, which is a picture right here, is actually one of the primary storage molecules of carbohydrates in humans. We actually will take glucose, and we take multiple, there's glucose, 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 and we store it together into this molecule of glycogen. Okay, carbohydrates are just rings of carbon. That's where carb comes from, attached to hydroxyl groups, which have an oxygen, that's the O, and then the hydrogen or the hydrate right there. Okay, mono, we have monosaccharides is what we would call them. Monosaccharides are good for energy. We have glucose and fructose in that category. We have disaccharides, which are going to be sucrose and lactose, and polysaccharides is how we're going to store them. So again, glycogen for animals, but cellulose for plants and also starch for plants. Cellulose is just like if you think about that stringy texture of celery, that is cellulose. Peptides or proteins, say these are amino acids, and what happens is we get these amino acids in their primary structure. Here you can see them in this chain, okay? And what happens is they start to sort of fold over like a curly ribbon idea, okay? And they start to form bonds, hydrogen bonds. They fold again into a tertiary structure, and then when we combine a bunch of them together, we get their quaternary structure, and that's where they're going to be both functional and structural. Proteins are both functional and structural. They're formed by the combination of just about 20 different amino acids. Some of them are found within our body. Our body can make them, which would mean that they are non-essential because our body already makes them. And then we have essential amino acids, and essential amino acids we have to eat within our diet. These are nucleic acids, and again, nucleic acids are going to be found in nucleotides as far as our genetic material. We also, ATP, for instance, is a nucleic acid. We can use it to store energy. Nucleotides are going to have a nitrogen base, a pentose sugar. Pentose has to do with the shape, a phosphate functional group, and they're going to help to transfer energy for our body functions as well as our genetic material. So let's talk a little bit about acids and bases. An acid is actually going to release hydrogen ions when it's dissolved within water, whereas a base is going to release hydroxyl ions when it's dissolved in water. So the big difference is it going to give us hydrogen ions, or is it going to give us hydroxyl ions? What kind of solution is it? So pH is a measure of the amount of hydrogen ions. pH stands for per hydrogens, or percent hydrogen, and it's the negative base 10 logarithm of the hydrogen ion concentration of the solution. Our pH scale is going to measure from 0 to 14. A pH of less than 7 is considered acidic. It goes from A to B, so less than 7 is acidic, 7 is considered neutral, greater than 7 is going to be basic, so A to B. So some important pHs within the body. Stomach acid has a pH of about 1. That is a very strong acid. Okay. Battery acid is going to be a 0. Lemon juice, gastric acid, vinegar, etc. is going to be about a 1. Okay. The blood pH, again, is on average about 7.4. Remember that range is 7.35 to 7.45. And then bile, which helps with digestion, also seawater, is about an 8. Okay. So this is just to give you some ideas of where we are in the pH range. So each pH unit is going to be 10 times more acidic, or has 10 times more hydrogen ions when moving from 14 to 0. So for example, moving from our pH of 6 to a pH of 3, there's 3 there, and so 3 and then 10 times that, so 10 to the third power is 1,000. Moving from pH 7 to pH 9 means the hydrogen ion concentration has decreased 100 times because 10 to the 2 second power is 100. Okay. Our blood pH of blood ranges from 7.35 to 7.45. Remember that average is 7.4. Enzymes, remember we talked about enzymes, that's where we're going to have that substrate coming and meeting and joining together. When our pH goes outside of that, our enzymes won't work with outside of that range of pH. And so if our enzymes aren't working, then our body's not functioning at its top level. So our body actually can use buffers to resist change within a pH. A buffer is a solution of a weak acid with what's called a conjugate base, so it's sort of matching base. That conjugate base can actually neutralize the hydrogen ions of the strong acid, and it turns it into a weaker one. The main buffer used in our blood is called bicarbonate. Bicarbonate. Remember that because it's going to come back to us in a little bit. Inorganic compounds do not contain both carbon and hydrogen, so water, salts, and some acids and bases are going to be used as inorganic compounds within our body. Okay, and so we can actually see that sodium here is actually used to transmit signals. This is how our body actually transmits signals, is using sodium. So respiratory acidosis is a condition that occurs when our lungs can't remove enough of the carbon dioxide produced by the body. Excess carbon dioxide can cause the pH of our blood to decrease, meaning that we're going to become acidic. Okay, so we decrease that pH, we become acidic, and that's because we can't remove the carbon dioxide from our lungs. So we get what's called respiratory acidosis. This will also often happen during any sort of airway obstruction, COPD, pneumonia, ARDS, even COVID, for instance. So we're kind of in that COVID crisis right now. Well, not kind of, but we are. And so when that happens, we could potentially have respiratory acidosis. So this can cause drowsiness, dizziness, disorientation, muscle weakness, hyperreflexia, rapid shallow respirations, decrease in blood pressure. The skin can become cyanotic or pale. Cyanotic means blue. We can get a headache. We can have what's called hyperkalemia, which is an increase in potassium. And that increase of potassium can cause a dysrhythmia within our heart. Okay. So make sure that you review these objectives, that you can identify the basic atomic structure, the major elements, and the difference between ions and atoms, that you can identify how atoms bond and the transfer of energy using ATP, that you can distinguish between organic and inorganic molecules and the biological significance of each group, and that you can describe respiratory acidosis and the relationship of molecules within the body. If you have any questions, you know where to reach me. I am happy to help you at any point. Thank you.